My lab is called the Underwater Acoustic and Navigation Laboratory. We develop signal processing solution for detection, classification, and really to deep understand everything below sea. We use sound as our eyes in the water and develop real data analysis tools to explore the marine environment.
We want to help the farmers to make better decisions on irrigation and fertilization. The objective of my studies is to understand how much to irrigate and when to irrigate. We are using numerical models to solve the water flow and solid transport in the soil-plant atmosphere continuum. We are also using sensors in order to get data from reality. This data is helping us to validate and calibrate the models with sensitivity analysis and also optimization tools.
Using a neural net, we designed an optimal optical system for observing volumetric microscopy data, and we used it to image subcellular structures in 3D at super resolution. The idea is to image dynamic structures, subcellular structures that are also changing in time, and to get a super resolution movie in real time.
By listening to the sea, we study the language of marine mammals like dolphins and sperm whales, and detect shipping activity to monitor noise pollution. Using advanced data analysis and machine learning, we analyze sonar signals to identify objects in the sea, like sea turtles and sharks, and to keep track on the marine environment. We also identify the existence of scuba divers and submerged mine for security application, and analyze data from marine observatories to answer present climate change questions, such as how much the level of the sea temperature increases, and our ocean current is changing.
One very powerful combination that we found is the combination of high-throughput microscopes with AI, where the idea is to exploit the high-throughput capabilities of the microscopes to produce the training data that would then be used to improve them.
I like to work on non-invasive tools. Two projects that we recently ended are working with MRI, magnetic resonance imaging, in order to describe the inside of a root system of tomato plant. We want to describe how sodium is penetrating the root system and how the sodium is accumulating near the root surface. Another topic that we recently ended the project is about date palm trees. We are using ERT, electrical resistivity tomography, that help us to explore the inside of the date palm tree and to explore the water flow inside the stem of this very important plant.
We highly believe in data-driven research, and there is much to explore at sea. We plan to combine our acoustics with marine robotics. For example, develop new sonar capabilities, design robust acoustic communication, and characterize the soundscape.
I'm working in Sde Boker campus, and there we have excellent facilities to conduct cutting-edge research. We have growth rooms, we have greenhouse conditions, and in those places I can conduct cutting-edge research for irrigation studies. I can manipulate the soils, I can manipulate the climate and also the plants in order to have best conditions for the experiment.
Microscopes are going to become even more clever. For centuries, microscopes have been developed, optimized for the human observer to produce the sharpest image. But recently, computers have been replacing humans also as microscope observers, and this means that the rules have changed. This combination between AI and microscopy is going to give us the next generation of bioimaging, enabling us to explore life in detail that was never possible before.